Hello again, BIO178. In this video, I'll walk us through some important points of our summary table to consider. First, what I'm going to show here is an example table that is provided in the publication associated with this diversity package. So I'll post this on Canvas if you're curious. But this table here is what the output is going to look like for that Excel file that we created by running that div basic command in R. So in these tables, each block here corresponds to a different population. So we're going to have two different blocks because we treated the 3 northern sites as one population and the 3 southern sites as another population. Each column, you're going to have one column for each of the different loci, in our case, the different SNPs that were included in the data set. So we would have 8 different loci and then an overall column at the end on the far right-hand side. And then each row here each row for each population gives us some different information. So if we scroll down to the caption for their figure, it gives us information about what we're looking at. So N here is the number of individuals per population sample genotyped per locus. So it's basically telling you for each of the different loci in your data set, how many individuals were actually genotyped for that particular locus. The next row, A here, is the total number of alleles per population sample per locus. So this is saying overall, how many different alleles did we identify at that particular locus? Don't worry about this percentage one. Let's go ahead and skip to allelic richness, which is something that we talked about in lecture, right? So both the A and the AR allelic richness are based on the alleles that are present in our samples. And this can be dependent on the type of marker you're using, right? So this example here, they used microsatellite markers. And if you look at the total number of alleles observed per population sample per locus, you can see that there's a big range from 2 to 14, for this population from 2 to 20. And then if we look at the allelic richness, it ranges again from 2 to about 17. So it's a relatively big range when we're looking at this data set, which was, again, based on microsatellite markers. Let's compare that to our data set. So remember when we executed that command, what we did is create an Excel file. So if we go to our folder, which should be Fall 20 RStudio Lab on our desktop, there should be now a folder corresponding to your data from that R function you ran. So if you open that, which I have already done, this is what our data looks like. So again, to orient us, we have this top block here is for the northern grouping. This bottom block is for the Southern California grouping. And if we remember first, we're looking at the allelic related metrics compared to that example data set. How does our data differ and why might that be? Think about what types of marker we were using compared to what type of marker they were using. So in this video, I'm going to point you in some directions similar to what the written instructions say about what I want you to think about. And these questions I'm asking in particular are for us to focus on not so much how to calculate the statistics, right? So all you had to do was punch in a function in R, but what I want us to focus on is how do we interpret that data and make sure we're understanding which one makes the most sense for our particular data. This function is basically giving us everything, but we want to think about what is it actually telling us and which of those is most appropriate and most informative for our actual data set. So that's what this part of the lab is focused on. So first of all, think about that. Compare the alleles and allelic richness from our data set to that example data set, which I'll also provide, and think about why they might be different. Another thing we want to consider is the sample size. So here we have the individuals that were sampled for each of these different loci across the different population groupings. Remember, we talked about in lecture, there are some metrics that are more appropriate than others if you have sampling size differences. Some metrics are sensitive to sampling size and some are not. So if we think about the next category of genetic diversity metrics based on heterozygosity, for instance, here those would be observed heterozygosity, that's this row, and expected heterozygosity here. Think about, based on what we talked about in lecture, which of those might be appropriate for this particular data set based on whether the sampling numbers are exactly the same for both populations across these different loci. So that's another thing to keep in mind. All right, another thing we want to think about is, based on whichever heterozygosity metric you think is most appropriate for this particular data set, are one of these two populations more susceptible or more of a concern in terms of long-term survivability in a changing environment? And it's hard to gauge that in an absolute sense, because actually genetic diversity metrics can vary across different species. So in this case, I want us to think about just in a relative sense. If you're just looking at these two populations, in terms of their heterozygosity and their genetic diversity, would one of those two populations be more of a concern for conservation purposes than the other? Yes or no, and why? Why do you think that? All right. And then the last thing is this column at the bottom, which is looking at what it's giving us is the p-value for our Hardy-Weinberg equilibrium chi-squared test. So remember, if the p-value is less than 0.05, that means the test is significant, meaning the observed and expected heterozygosities we see are significantly different. That means if your p-value is less than 0.05, that particular locus in that population is not in Hardy-Weinberg equilibrium. And remember, in lecture, we said oftentimes that points to actually something pretty interesting. So what I want you to do is look at this bottom column for both populations across the different loci, the different SNP loci, and see if there are any loci here that are out of Hardy-Weinberg equilibrium, and think about why that might be the case. I'll note in particular that this locus here is a little bit funky for Southern California. It looks different than the other ones. We don't actually have a p-value for our Hardy-Weinberg equilibrium chi-squared test. So look at the data, see how it's different, and try to figure out why that might be the case. It's actually pretty interesting. I will also say that we're going to revisit this data set a little bit later. So when you're looking at these Hardy-Weinberg equilibrium tests and thinking about why they might be out of Hardy-Weinberg equilibrium, I'll tell you that I specifically put within this subset data that we're looking at, 8 different loci, I actually put in some loci on purpose that I have found through other metrics, other statistical tests, which we'll talk about later. They are actually outliers. So I've done that on purpose for us to see what the data might look like if you're looking at overall neutral SNPs or maybe some adaptive SNPs as well. So those are some things to consider for this data set. Again, I'm specifically not telling you the quote-unquote answers, but I'm pointing you in the right direction of things I want you to consider based on what we talked about in lecture.